Welcome back to the Deep Dive. Today, we are tackling something that I really like to think of as the ultimate medical cheat code.
That's a good way to put it.
We've spent the last few sessions just wading through the chaotic, messy reality of how the human body fights infection. All that inflammation, the fever, the cellular warfare.
And collateral damage.
So much collateral damage.
Yeah.
But today we are flipping the script. We are moving from that reactive defense to a calculated, engineered defense. We are talking about vaccines.
It really is a massive shifting perspective, isn't it? I mean, up until now in our immunology arc, we've looked at the immune system as a responder.
Right. Something attacks
Yeah.
and the body just goes haywire.
Exactly. A breach occurs and the body responds, often violently.
Yeah.
Vaccines represent foresight. It's this amazing intersection of public health and molecular biology where we essentially trick the immune system.
How so?
We get it to create a memory file, but without all the trauma of the actual war, without the actual infection.
All the gain, none of the pain. Or at least significantly less pain than, say, getting actual smallpox.
Precisely.
And just to orient everyone, our roadmap for today is drawn primarily from Murray's medical microbiology. We're in Chapter 11. But as always, we're going to be looking under the hood.
We have to. We'll be using Jowette's, Melnick, and Edelberg's, mostly Chapter 8, to really understand the cellular mechanics that make these things actually work.
And for any student listening, why does this matter so much? I mean, beyond the obvious public health implications.
Well, because this is where it all comes together, and because exam boards absolutely love asking about the type of immunity.
Active versus passive.
And the mechanism of the vaccine. Live, killed, conjugate. If you can understand the biology, you can predict the clinical application. You can reason through it.
So you don't have to just memorize a list.
Yeah.
You can actually understand why one is used over another.
That's the goal. Understanding the why is everything.
Okay, so let's start with the big picture, the why. What is the strategic goal here? Beyond just preventing one person from getting sick.
The ultimate goal, the dream, is eradication. But to get there, we need to break the chain of transmission. And that brings us to this really core concept of herd immunity.
Herd immunity. I love that term. It sounds a bit, agricultural. But it's actually this incredibly sophisticated idea. It's the human shield concept, right?
That is a perfect metaphor. It's not just about your individual armor. Think of a virus like a forest fire.
Okay.
It needs to jump from tree to tree to keep going. If all the trees are dry and susceptible, the fire just rages out of control.
But if you can protect some of those trees.
Exactly. If you wet down a critical portion of those trees, if you immunize them, the fire hits a wet tree and it just stops. It has nowhere to jump.
It hits a dead end.
A dead end. So when a critical percentage of a community is immunized, the spread of the infectious agent is limited because there are just not enough susceptible hosts to sustain an outbreak.
So this is crucial for protecting the people who can't be vaccinated.
Absolutely critical. We have to remember, not everyone has a choice in this. You've got newborns who are too young for certain vaccines. You have patients who are immunocompromised.
People on chemotherapy, transplant recipients.
Great. They are the dry trees in the forest. They are relying on the herd. They are relying on the fact that the virus literally cannot find a path to them because everyone standing around them is immune.
And Murray points out some incredible success stories here. I mean, the gold standard has to be smallpox.
Smallpox is the only infectious disease of humans that has been eradicated worldwide. The last natural case was in 1977. It's just a monumental achievement.
Which is truly mind-blowing. We wiped an entire disease off the face of the earth. But why smallpox? Why haven't we done that with everything? Why do we still have the flu every year?
That's a great question, and it really comes down to the biology of the pathogen itself. Smallpox was, in a way, the perfect candidate for eradication.
What made it perfect?
Well, first, and this is the most important part, it has no animal reservoir. It only infects humans. So it has nowhere else to hide.
Nowhere. If you vaccinate all the humans, the virus is extinct. It can't go hang out in a bat or a pig and wait for us to drop our guard. Second, the symptoms are obvious. You get those pustules, you know who's sick, and you can quarantine them effectively.
And the vaccine was stable.
Very stable and very effective against the single serotype of the virus. Now, contrast that with something like the common cold, the rhinovirus.
Right, which has hundreds of different versions.
Hundreds of serotypes. You'd need this massive vaccine cocktail, and by the time you made it, the virus would have already mutated again. Or look at influenza.
It's got an animal reservoir, Birds, pigs.
Exactly. Even if we vaccinated every single human on the planet against the flu, the virus would just hang out in the livestock, reshuffle its genes, that whole antigenic shift and drift thing we talked about, and then jump right back over to us next year.
So eradication is incredibly difficult for those zoonotic diseases. Smallpox was kind of the low-hanging fruit, biologically speaking. What about polio? Murray mentions we're getting close. We are so close with polio. We've eliminated the wild-type virus in most of the world. It's another huge success story. But then you have the other side of the coin.
The failures, or maybe challenges is a better word.
Challenges is a good word. Measles is the prime example.
Measles just keeps popping up in the news, doesn't it? Even though we have a fantastic vaccine for it.
It does, and that is largely due to complacency and, frankly, misinformation. Measles is one of the most contagious viruses we know. It is a very high R0. It's a reproductive number.
So you need a really high vaccination rate to maintain that herd immunity.
You need something like 95% of the population vaccinated. If that number drops, even just a little bit, to say 90%, the virus finds the cracks in that shield.
It's a reminder of how fragile that protection can be.
It really is. Now, to achieve this herd immunity, we need the individual immune system to play ball. We need to create what Murray calls immunologic memory.
And this is where we bring in JVETs to look under the hood.
Yeah.
To really understand vaccines, you have to get the difference between a primary and a secondary response.
Absolutely. So let's walk through that. The first time your body sees a brand new pathogen, that's the primary response. And the key word here is slow.
How slow are we talking?
Days, sometimes weeks. Your body has to recognize this new antigen, find the right B cells that match it, and then start expanding them, cloning them. It's a whole process. And the initial antibody you produce is IgM.
IgM. I would think of M for mega because it's that big pentamer molecule. But it's also kind of a rough draft, isn't it?
It is. It has lower affinity. It's not as specific. It's good at grabbing things because it has 10 binding sites. It's like a big sticky net, but it's not the refined high-precision weapon you want.
And during this lag time, while the body is trying to ramp up, the pathogen is just replicating like crazy.
That's the window of disease. That's when you get sick. You get the fever, the tissue damage, all of it.
So the whole point of a vaccine is to hack that lag time.
Precisely. We want to skip that dangerous primary response entirely. A vaccine exposes you to the antigen, but under safe, controlled conditions. Your body goes through that learning curve. It builds up its army of memory B cells and T cells, all without the risk of the actual infection.
So that when the real virus shows up later,
you trigger a secondary response. And this is a whole different ballgame. It is rapid, it is robust, and crucially, it is dominated by IgG.
IgG. G4, good to go.
I like it. Let's go with that. IgG has much higher affinity. It binds tighter to the target. It gets into tissues better. And because you already have those memory cells just waiting, you produce massive amounts of it almost immediately.
So the virus is neutralized before it can even establish a foothold.
That is the entire goal, before it can even cause disease.
Okay, so we're preloading the immune system. But there are two very different ways to get these antibodies right. You can make them yourself, or you can just borrow them.
Right, and that's the core distinction between active and passive immunization.
I love the analogy here. Teach a man to fish is active immunity.
And give a man a fish is passive immunity. It's a perfect analogy.
So break that down. What are we doing in passive immunization?
We are literally injecting purified antibodies, or sometimes immune cells, into a patient. The patient's own immune system is doing absolutely nothing. It's just sitting back and watching.
So what's the advantage of that? I mean, if you're just giving someone a fish, it gets eaten and it's gone. Why would we ever do that?
The advantage is one thing: speed. Active immunity, teaching your body to fish, takes weeks to develop.
Right, that lag time we just talked about.
Exactly. But if you have a patient who has just been bitten by a rabid dog or stuck with a hepatitis B infected needle, you don't have weeks. You need protection now.
The virus is already in the race.
It's racing toward the central nervous system or the liver. You need to intercept it immediately. So passive immunity is a stopgap. It buys you time.
But it doesn't last, right?
It doesn't last. The half-life of an IgG antibody is about 21 days. Once those injected antibodies degrade and get cleared, the protection is gone. You're back to square one. You have no memory B cells of your own.
Okay, so Murray breaks this down into natural versus artificial. Natural passive immunity seems pretty straightforward.
Yeah, this is nature's original vaccination program. Maternal IgG crosses the placenta to protect the fetus.
So the baby is born with a starter pack of the mother's immunity.
A starter pack, exactly. It lasts for the first few months of life. And then you also have IgA in breast milk, which coats the baby's gut and protects against enteric pathogens.
That's natural. Then we have artificial, which is therapeutic. This is where we get into that alphabet soup of immune globulins.
Right, so we can use human immune globulin, which is just pooled plasma from thousands of blood donors. It contains a random sampling of whatever antibodies are floating around in the general population.
And what would we use that for?
General prophylaxis for something like hepatitis A, or for treating patients with immunodeficiencies who can't make their own antibodies at all.
But sometimes you need more than a random sample. You need the special forces.
That's where specific high-titer globulins come in. These are harvested from donors who we know have super high levels of antibody against one specific disease. And for exams, there's a big 5 that you really, really need to memorize.
Let's run through them, the big 5.
Okay, first, HBIG, hepatitis B immune globulin, for needle stick injuries or for newborns born to Hep B positive moms.
Got it, HBIG.
Second, VZIG, varicella zoster immune globulin. You'd use this for an immunocompromised kid who gets exposed to chickenpox.
Okay, VZIG.
Third, RIG, rabies immune globulin. This is absolutely critical for post-exposure prophylaxis. You inject it right into and around the wound.
RIG, makes sense.
Fourth is TIG, tetanus immune globulin, for that deep, dirty wound from a rusty nail. And fifth is CMV globulin, mostly used for transplant patients.
HBI, VZIG, RIG, TIG, CMV. Okay, I'm locking those in. Now, Murray mentions a risk here. Historically, we sometimes use horse serum for this.
Yes, equine serum. And while it works, an antibody is an antibody, structurally, but the human immune system is smart. It looks at the constant region of that horse antibody and says, hang on, that's not one of ours.
So your body attacks the cure.
Essentially, yes. It triggers an immune response against the foreign antibodies, and this leads to something called serum sickness.
Serum sickness, that sounds unpleasantly Victorian.
It's a classic type 3 hypersensitivity reaction. Your body makes antibodies against the horse antibodies. They bind together and form these big immune complexes.
And those just get stuck everywhere.
They get stuck everywhere. They clog up the filters in the kidney, causing glomerulonephritis. They get deposited in the joints, causing arthritis. In the blood vessels, vasculitis. It's a systemic problem.
So, bottom line, prefer human if you can get it. Passive is fast, temporary, borrowed. Now let's move on to the main event, active immunization. This is where we're trying to get the body to make its own memory.
Right, and Murray splits this into two massive categories, inactivated and live.
Let's start with inactivated. These are the safe but clunky options, as you put it.
That's a good way to think about them. You're taking the pathogen and you're killing it with heat, with chemicals like formalin, with radiation. Or you're just taking a piece of it, a subunit.
So by definition, it cannot replicate in the body.
Correct. And that's the huge safety factor. A dead bug cannot cause disease. It cannot revert to a virulent form. It's very safe for immunocompromised patients, for pregnant women, for everyone.
But because it's dead, it's also kind of boring to the immune system, isn't it? It doesn't seem very threatening.
That is the central problem of inactivated vaccines. Because there's no real infection, there's no intracellular replication, the immune system just sees it as some random debris floating by. In Jawlit's terms, this is an exogenous antigen.
Okay, let's trace that pathway. Exogenous antigen means it's coming from outside the cell.
Right. So it gets eaten by an antigen-presenting cell like a macrophage. It gets taken into a little bubble of phagosome chopped up into pieces. And here's the key part. It gets loaded onto MHC class II molecules.
MHC class II, the pathway for outside stuff.
Yep. And MHC2 presents to CD4 helper T cells. Specifically, it tends to drive a Th2 response, which leads to B-cell activation and antibody production.
So you get antibodies, you get a humoral response.
You do. But what you don't get is a good cell-mediated response.
Yep.
You get very few of those CD8 killer T cells because the antigen never actually got inside the cytoplasm to trigger the MHC class I pathway.
So we have a vaccine that's very safe, but produces a weaker, less complete response. How do we fix that? How do we make the immune system actually care about this dead debris?
We need to give it a hype man. We need an adjuvant.
The adjuvant, the ingredient that basically slaps the immune system in the face and tells it to wake up.
That's a pretty good description. The most common one you'll see is alum various aluminum salts.
And how does aluminum actually help?
It works in two main ways. First, simple chemistry. It causes the protein antigen to precipitate, to clump up into particles. This makes it much, much easier for macrophages to see them and eat them compared to a tiny soluble protein just floating by.
Okay, so it makes it a bigger target.
A bigger, tastier target. And second, and this is more important, it irritates the local tissue. It triggers the innate immune system. It acts as a danger signal that activates toll-like receptors and the NLRP3 inflammasome.
So it's mimicking the danger of a real infection, even though there isn't one.
It's tricking the body into thinking there's a threat, which ramps up the whole response. Without the adjuvant, the immune system might just ignore a purified protein. With the adjuvant, you get inflammation, you get cytokine release, and you get a robust immune response.
Okay, so inactivated vaccines need adjuvants. They often need boosters. Murray lists a few different types. Let's start with killed whole cell.
These are just what they sound like. The Salk polio vaccine, or IPV, is a classic example. Also hepatitis A, and the rabies vaccine. The whole virus is there, it's just been chemically nuked, so it's dead.
Then we have toxoids. This is a really crucial distinction for things like diphtheria and tetanus.
Yes, this is a key conceptual point. For those bacteria, Corynebacterium diphtheria and Clostridium tetani, the bacteria themselves aren't the main problem. It's the incredibly potent toxin they produce that causes all the disease.
So the vaccine doesn't even contain the bacteria.
Exactly. It contains the toxin, which has been chemically modified so it's no longer toxic. We call that a toxoid. You're immunizing against the weapon, not the attacker.
So your antibodies are there to neutralize the toxin before it can do any damage.
Precisely. And finally, you have subunit vaccines, just taking one specific piece of the pathogen.
Hepatitis B is the classic example here, right?
It is. We use the hepatitis B surface antigen, HBsAg, and what's really cool is we don't even have to grow the virus. We clone the gene for that protein into yeast cells, and we turn the yeast into little factories that pump out the surface protein for it.
A recombinant vaccine. Very clever. HPV is similar.
Very similar. The HPV vaccine uses the L1 protein, which has this amazing property where it self-assembles into virus-like particles or VLPs. They look exactly like the real virus on the outside, but they're completely empty on the inside. No DNA, no risk of infection.
But the immune system sees it and thinks it's the real deal.
It thinks it's the real deal. Now, we have to pause here for what Murray rightly calls out as a super high-yield topic, the sugar problem.
Ah, yes. Polysaccharide versus conjugate vaccines. This comes up all the time.
It's a massive differentiator on exams and in clinical practice. So you have these encapsulated bacteria, Haemophilus influenza type B, or Hib, meningococcus, pneumococcus.
They all wear this thick sugar coat, a polysaccharide capsule.
And that capsule is their stealth cloak. It helps them evade phagocytosis. So to fight them, we need antibodies that can stick to that sugar. But there's a big problem.
What's the problem?
Sugars are T-independent antigens.
T-independent, meaning the T cells just completely ignore them.
They are total protein snobs. They only recognize peptides presented on MHC. They cannot see sugars. So when a B cell sees a sugar capsule, it can get activated a little bit on its own. It'll make some IgM.
But without the T cell.
Without that T cell helper, you get no class switching to IgG. You get no affinity maturation to make the antibodies better. And critically, you get no memory. It's a weak, short-lived, forgettable response.
And there's a specific group of people who are especially bad at this, right?
Infants. Children under 2 years old have a very immature T-independent response. Their B cells just aren't equipped to handle sugars on their own. If you give a pure polysaccharide vaccine to a 6-month-old, it does almost nothing.
Which is why the old pneumococcal vaccine, Pneumovax 23, is only for adults.
Exactly. But we desperately needed to protect babies from meningitis and pneumonia caused by these bugs. So we had to engineer a solution. We had to find a way to trick the T cells into helping out.
Enter the conjugate vaccine.
The solution. The word conjugate just means we chemically link the sugar to a protein, usually something the baby's already being vaccinated against, like diphtheria toxoid.
And how does that trick work? It sounds almost like a Trojan horse.
It's a brilliant Trojan horse mechanism. So the B cell has a receptor that recognizes and binds to the sugar on the outside. That's its target.
Okay.
It binds the sugar and pulls the whole complex inside the sugar and the protein that's attached to it. Now, inside the B cell, it chops everything up. And it takes the protein part of that conjugate
And puts it on its MHC class 2.
Exactly. So now the B cell is waving this little peptide flag from the protein. A helper T cell comes along, sees the protein flag, and says, oh, a protein peptide. I know how to deal with that. I can help you.
Even though the B cell is really trying to make antibodies against the sugar,
The T cell doesn't care. It activates the B cell, releases cytokines, and gives it the signal to go through the whole process. Class switching, affinity maturation, memory.
So you get a powerful, long-lasting IgG response
Mm-hmm.
against the sugar.
All because we smuggled a protein in the back door. This is why the Hive vaccine virtually wiped out Haemophilus meningitis in kids, and why Prevnar, the pneumococcal conjugate, is a cornerstone of the childhood vaccine schedule.
So if you see an exam question asking why we give a conjugate vaccine to a baby, but a plain polysaccharide vaccine to an adult,
The answer is always, babies cannot mount an effective response to T-independent polysaccharide antigens. They need the protein conjugate to recruit T-cell help.
that is just brilliant. Okay, let's pivot. We've covered the safe and activated stuff. Now let's talk about the other side of the coin. The high-risk, high-reward option, live attenuated vaccines.
These are the heavy hitters. MMR for measles, mumps, rubella, varicella for chickenpox, rotavirus, oral polio. In these vaccines, the pathogen is still alive. It can still replicate.
But it's attenuated, weakened. How do they actually do that? Do they just, I don't know, send it to the gym less often?
Something like that. Murray describes the classic process. You take a human virus and you grow it in something that isn't human, like monkey kidney cells or in chick embryos. Or you grow it at a lower temperature than the human body.
And over time, it just adapts to that new environment.
It adapts through random mutation and selection. It gets really good at growing in monkey cells, but it loses the specific genes that made it good at infecting humans at 37 degrees Celsius. It becomes a wimp in our bodies.
So when you inject it into a person, it sets up a real but very mild infection.
A very mild, controlled infection. It replicates a little bit, and that's the key.
Why is that so much better?
Because it mimics the natural infection almost perfectly. Remember our pathways. This is a live virus that is getting inside the cytoplasm of your cells. That means it enters the endogenous pathway.
Endogenous pathway means MHC class 1.
MHC class 1, which presents two CD8 cytotoxic T cells, your killer T cells. So with a live vaccine, you get the full orchestra. You get the humoral immunity with antibodies, and you get the cell-mediated immunity with killer T cells.
And the immunity is often lifelong after just 1 or 2 doses.
Exactly. It's a much more potent and durable teacher for the immune system, but, as you said, high risk.
So what's the dirty laundry? What are the disadvantages?
Well, first and foremost, because it is a live replicating organism, you do not give it to immunocompromised patients or pregnant women. A weak virus can still cause devastating disease in a weak host.
That is a hard and fast rule for exams, a huge contraindication.
Absolutely. Second, there's stability. Live organisms need to stay alive. That means they require a cold chain, constant refrigeration from the factory all the way to the clinic. That's a huge logistical challenge in many parts of the world.
Much harder than a freeze-dried killed vaccine.
Much harder. And third, there is the nightmare scenario, reversion.
The virus mutating back to its original dangerous form.
It's rare, but it can happen. The vaccine virus replicates in your body. Every replication is a chance for a mutation. If it gets a lucky mutation, it can regain its virulence. And this is the central story of the oral polio vaccine.
Okay, let's do that case study. Polio is the ultimate showdown of these two vaccine philosophies. Salk versus Sabin.
It's a fantastic historical comparison. So Jonas Salk developed the IPV, the inactivated polio vaccine. It's a killed virus given as an injection in the arm.
So because it's injected, it stimulates a strong IgG response in the blood.
A very strong serum IgG response. So if the poliovirus ever gets into your blood, those IgG antibodies will neutralize it immediately. It can't reach the spinal cord. It prevents you from getting paralyzed.
So it protects the individual perfectly. But there's a catch, isn't there?
There is. IgG in the blood doesn't really get into the gut very well, and polio is a fecal oral virus. It replicates in the intestines.
So if I'm vaccinated with Salk, I won't get paralyzed, but I can still get a gut infection.
You can. You won't feel sick, but the virus will replicate in your gut and you will shed it in your feces. You can still pass it on to an unvaccinated person. So Salk protects you, but it doesn't stop transmission.
Okay, so then along comes Albert Sabin.
Albert Sabin develops the OPV, the oral polio vaccine. This is a live attenuated virus given as drops on a sugar cube.
Oral. So it's going right to the site of infection.
Right to the gut. It replicates in the lymphoid tissue there. So yes, it stimulates serum IgG just like Salk, but crucially, it also stimulates a powerful mucosal IgA response.
IgA, the bouncer at the door of the mucosal surfaces.
The bouncer at the door of the gut. That IgA stops the wild poliovirus from ever even establishing an infection in the first place. So Savin's vaccine prevents paralysis and it prevents transmission. It breaks the chain. This is why OPV was the tool for global eradication. It creates herd immunity much more effectively.
But there's that trade-off, the risk.
The trade-off. Because the Sabin vaccine is live, it replicates. And in about 1 in every 2.4 million doses, the vaccine virus reverts and causes vaccine-associated paralytic polio, VAPP.
1 in 2.4 million. That is a terrifyingly small but real number.
It is. Back when wild polio was paralyzing thousands of kids every year, that risk was absolutely acceptable. But in a country like the U.S. where we haven't had wild polio for decades, that risk is no longer acceptable.
So that's why we switched.
That's why the U.S. switched exclusively to the Salk vaccine, the IPV, in the year 2000. We used the killed vaccine to maintain individual protection without any risk of reversion.
It's a fascinating example of balancing risk versus public health benefit. Now let's look to the future, where he talks about genetic engineering, moving beyond just growing bugs and eggs.
We are definitely in a new era. The new frontier is all about recombinant and nucleic acid vaccines.
You mentioned recombinant subunits like Hep B, but there are also hybrid viruses.
Right. Imagine you take a safe, harmless virus like canarypox, which can infect human cells but can't cause disease, and you use genetic engineering to splice in a gene from a really bad virus, like Ebola.
So the canarypox virus acts like a delivery truck.
It's a delivery truck. It infects your cells, delivers its package, the Ebola gene. Your cells express that protein, and your immune system learns to fight Ebola without ever seeing the actual virus.
That's brilliant. But the real revolution has been DNA and RNA vaccine.
Oh, absolutely. The topic of the decade. Conceptually, it's just so elegant. Instead of injecting the protein antigen itself, we inject the instruction manual.
The DNA plasmid or the mRNA strand that codes for the protein.
Yes. And your own muscle cells at the injection site take up that genetic code. They read the instructions, and they become the vaccine factory. They produce the viral protein inside their own cytoplasm.
And because it's being made inside the cell.
It enters the MHC class 1 pathway. This is the holy grail. You get the endogenous presentation and the CD8 killer T cell response that you normally only get with a live vaccine. But there is no live virus, there's no risk of infection, no risk of reversion.
It's the safety of a killed vaccine with the power of a live one.
That's the promise.
There's one more cool concept Murray mentions called reverse vaccinology. What's that about?
This is using big data instead of petri dishes. For meningococcus type B, for a long time we couldn't find a good vaccine target. The sugar capsule looked too much like our own human antigens.
So what did they do?
They sequenced the entire bacterial genome. Then they used a computer to run an algorithm to predict every single protein the bacteria could possibly make that might be on its surface.
So they found the targets on a computer screen first.
They found them in silico. They identified the most promising candidates, synthesized just those proteins in the lab, and tested them. And that's how the MenB vaccine was created. It was born in a computer.
That is just incredible. Okay, we've covered a ton of biology. Let's try to consolidate this for a student in the clinic. What are the key rules of the road?
Okay, let's do a quick clinical summary. Rule number 1, timing matters.
How so?
We don't give live vaccines like MMR or varicella to newborns. That's because the mother's IgG is still floating around in their system from the placenta. If you inject a live measles vaccine into a 2-month-old, mom's antibodies will just kill it before the baby's immune system has a chance to learn from it.
Which is why we wait until they're about 12 months old for those.
Exactly. Rule number 2, the site dictates the response. If you want gut immunity, that secretory IgA, you need a mucosal route, like the oral polio or rotavirus vaccines.
And injected vaccines give you systemic IgG.
Mostly. And rule number 3 is just the public health balance. It's always a trade-off between efficacy, where leave is best, versus safety, where killed is best, versus cost and stability.
And of course, rule number 4, know your patient, immunocompromised, pregnant, no live vaccines.
That's the big one, always.
All right, I think it's time. Clear your desks, everyone. We are going to do a rapid recall SBA drill. Single best answer. I will ask the question, I'll pause for a moment, and then our expert will break down the answer and the why.
Let's do it.
Question 1. A 4-month-old infant is brought to the clinic for routine vaccinations. You need to protect them against Haemophilus influenza type B, Hib. You have 2 options, a pure polysaccharide vaccine or a polysaccharide protein conjugate vaccine. Which formulation is required to elicit a protective memory response in this patient? Pause.
The answer is the conjugate vaccine. Here's why. The patient is an infant, under 2 years old. As we discussed, their immune system cannot respond effectively to T-independent polysaccharide antigens, so the pure polysaccharide vaccine would be useless. By conjugating or linking the sugar to a protein, we recruit T-cell help via the MHC class II pathway. This allows for class switching to IgG and the development of immunologic memory.
Perfect. Question 2. A 30-year-old spelunker is bitten by a bat while exploring a cave. To provide immediate protection against rabies, which agent should be administered at the wound site? Pause.
The answer is rabies immune globulin, or RIG. The key word in the question is immediate. The patient has already been exposed. We cannot wait weeks for an active immune response to develop. We need to give them passive immunity right now. We inject the preformed antibodies, RIG, directly into and around the wound to neutralize the virus before it can enter the nerves. We do also give the inactivated rabies vaccine at a different site to build long-term immunity, but for immediate protection, it has to be RIG.
Excellent. Question 3. Which specific vaccine type is absolutely contraindicated in a pregnant woman due to the theoretical risk of fetal infection? Is it A, tetanus toxoid, B, inactivated influenza, C, MMR, or D, hepatitis B subunit? Pause.
The answer is C, MMR, which is a live-attenuated vaccine. The rule is simple: no live vaccines in pregnancy. There is a risk, however small, that the weakened virus could cross the placenta and infect the developing fetus. In fact, the others listed are not only safe, but often recommended during pregnancy.
Great point. Question 4. Which adjuvant is most commonly used in inactivated human vaccines, like the hepatitis B vaccine, to enhance their immunogenicity? Pause.
The answer is alum, which is aluminum hydroxide or aluminum phosphate. It's the classic, most widely used adjuvant in human vaccines. It works by precipitating the antigen and by triggering the innate immune system.
Last one. Question 5. A patient receives the Salk polio vaccine, the IPV. Months later, they ingest wild poliovirus from contaminated water. Which of the following best describes the most likely outcome?
The answer is that the virus will likely replicate in their gut, but it will not invade the central nervous system to cause paralysis. Remember, the Salk vaccine is injected and produces systemic IgG. That IgG is great at protecting the blood and nerves, so no paralysis. But it does not produce good mucosal IgA in the gut. So, the virus can still set up a local infection in the gut and be shed, potentially infecting others, even though the vaccinated individual is protected from disease.
Fantastic. Well done if you get those right at home. If not, this is a great reason to go back and really solidify those mechanisms.
Definitely. And crack open Murray, Chapter 11. Specifically, look at Figure 11.1. It has this beautiful visual flow chart of all the vaccine types we talked about. It really helps to have that mental image.
So let's wrap this up. We have engineered immunity. We learned that passive is borrowing. It's fast, but temporary.
It's giving a man a fish.
Exactly. Active is owning it, teaching the immune system to fish for itself.
Killed vaccines are the safe and reliable option, but they need that adjuvant hype man to get noticed.
Live vaccines are the drill sergeants. High risk, high reward. They mimic the real war to create the best memory.
And conjugates are that brilliant trick. The way we protect babies by disguising sugars as proteins so the T cells will pay attention.
It's just such a powerful demonstration of applying basic immunology to solve a massive public health problem.
It really is. And you know, vaccines are unique in medicine. They're the only drug where the ultimate measure of success is to stop using them. The goal is to put the manufacturer out of business by completely eradicating the disease. We did it with smallpox. We are so close with polio.
That is a powerful thought. Engineering our way out of a job in the best possible way. So here's a final thought for you to chew on as we close out. We've been talking about vaccines for infectious diseases, but what about vaccines for things that aren't infections?
Where are you going with this?
We're now seeing research into vaccines for cancer, training the immune system to attack tumor cells, vaccines for Alzheimer's, to clear amyloid plaques. There are even experimental vaccines for addiction, training your body to make antibodies that bind to cocaine molecules in the blood so they can't reach the brain.
It's applying the same fundamental principles to a whole new set of problems.
Exactly. The principles we talked about today, adjuvants, antibody affinity, T-cell help, they have the same principles that might one day treat our most challenging chronic diseases. The immune system is a tool. We're just beginning to learn how to wield it.
A very powerful tool.
On that note, stay immunized, stay curious, and we will see you on the next Deep Dive.